Hello students, today we'll discuss one of the most important topic of molecular basis of inheritance of 12th, that is here the central dogma of molecular biology. See, what is central dogma? Before knowing all these things, you have to be very much clear about the structure of DNA, what I have also taught you, the composition of nucleotides, all these things. But one thing to make you sure that DNA is made up of what? Means the nucleotides, they make the nucleic acid, but DNA is the genetic material. So genetic material means it is made up of the structural and functional units called genes. So what are these genes, where these are present, how these are arranged inside the genome, or how these are arranged inside the DNA. Okay, so and what about the function of a gene? Gene is the structural functional unit. So a single gene, it synthesizes either mRNA or tRNA or rRNA. Means what is there? Different genes are there. Not all the genes they code for the proteins. Only the protein coding genes they synthesize the mRNA, and other genes they synthesize tRNA or rRNA. Okay, so the process, what happens, DNA can make an exact copy of itself, that you must knowing, that is DNA replication. DNA replication means DNA, it makes the exact copy, it doubles inside the cell, that is DNA replication. DNA also synthesizes all the three different types of RNAs, that is mRNA, tRNA and rRNA. mRNA is the messenger RNA. Messenger means what? It carries the message. What type of message it carries? It carries the message of genetic information. It carries the message of what? Protein. Means how the protein is to be synthesized. Which amino acids they form the protein. In which sequence the amino acids to be localized. All these things are present inside the mRNA. So it's called the messenger RNA. Because from a single gene, it carries the message for the synthesis of a particular or specific protein. Okay. So what is the thing? Gene, it synthesizes mRNA or tRNA or rRNA. All these three different types of RNAs, they have a combined role for the synthesis of a protein. Okay, so DNA, it synthesizes DNA, that is replication. DNA synthesizes all the three types of RNAs, that is called transcription. And mRNA, it transcribes, means mRNA, after the transcription process is over, with the help of tRNA and rRNA, it translates the protein, means protein synthesis occurs. Okay, so what is the role of tRNA? mRNA is the messenger. Then tRNA, what is its role? It transfers the amino acids from the cytoplasm according to the codon sequence of mRNA. What is the codon sequence? Codon sequence means there is the triplet nucleotide sequence which are present in a sequence inside the mRNA. And each codon, it codes for a particular amino acid. So according to the codon sequence of a particular mRNA, different amino acids, they are brought to the ribosome. Ribosome is the protein factory. There the amino acids, they are brought by tRNA. So the name tRNA, that is transfer RNA, is given to it. Okay, it transfers the amino acids from the cytoplasm to the protein synthetic site, that is ribosome. Okay, so what is the role of rRNA then? rRNA means it is ribosomal RNA. It is present inside the ribosome. And as I have told that ribosome is the protein factory, and ribosome is made up of rRNA and proteins. Okay, so these rRNAs, they have also a specific role for the protein synthesis, that is translation. What they do? These rRNAs, they help for the binding of mRNA with the ribosome during protein synthesis, and binding of tRNA along with the amino acid during protein synthesis. So all these three RNAs, mRNA, tRNA and rRNA, they combiningly are used in the process of translation. Okay, so all these things are clear now. So the process of synthesis of RNA is called transcription, and the process of synthesis of protein from mRNA is called translation. So what is central dogma? Central dogma is the one-way flow of genetic information from the DNA to the protein. Means how a particular protein is synthesized. Maybe it may be a structural protein or an enzyme, but how it is synthesized, which kind of gene it synthesizes, all these things, that is a one-way flow of genetic information. Okay, from gene to mRNA and mRNA to protein. This one-way flow of genetic information is called central dogma, and this word central dogma, it was coined by Francis Crick. Okay, so let's discuss all these things now in details. See, DNA, it can replicate itself, that is called replication. It can make an exact copy of itself. So DNA also can synthesize mRNA, tRNA and rRNA. mRNA is the messenger RNA, just we have discussed. So mRNA, it carries the message for the synthesis of the protein. Okay, mRNA along with the help of tRNA and rRNA, it synthesizes the protein. So the process of synthesis of all the three types of RNA from DNA is called transcription, and the process of synthesis of protein from mRNA is called translation. So this is the one-way flow of genetic information from DNA through the RNAs into the protein. So this one-way flow of genetic information is called central dogma of molecular biology. This central dogma of molecular biology term, it was proposed by Francis Crick. Okay, and it was made confirm that the information, the genetic information or the information for protein synthesis, it is only in one way direction. Okay, it was made by the, it was also received by many of the scientists. Okay, later on what happens, this central dogma concept, it was changed. Means what happens after the discovery of reverse transcription. What is reverse transcription? Reverse transcription, as I have told you, transcription is the process of synthesis of RNA from DNA. Reverse method means synthesis of DNA from RNA. It is called reverse transcription. So mRNA is always single stranded. I have also discussed all these things in my prior classes, that RNA molecules are always single stranded and are 5' to 3' directed. Okay, so these synthesis of what? Single stranded DNA. Is mRNA single stranded? It must be synthesizing a single stranded DNA only. So the process of synthesis of cDNA, that is called, cDNA means complementary DNA, that is single stranded. It is synthesized from mRNA. mRNA synthesizes a cDNA. cDNA is the complementary DNA, or it is also known as ssDNA, single stranded DNA. Okay, this ssDNA, it is synthesized from the mRNA. So this process is just the reverse of transcription and is known as reverse transcription. This process, it happens in case of retrovirus infection. Means what? Supposed to be any retrovirus, it infects any eukaryotic body. Then we know that in case of eukaryotes, the genetic material is DNA. Then how the retroviral infection occurs inside us? Because the genetic material of a retrovirus is RNA. Okay, so inside the eukaryotic cell, the mRNA, that is the RNA or the genetic material of the retrovirus, it synthesizes the single stranded DNA or complementary DNA, which again synthesizes its complementary strand of DNA. Means it is complementary, single stranded, it synthesizes its complementary copy. So they again wind upon each other to form a double stranded DNA. Now this double stranded DNA of retrovirus, it mingles with the host cell genome or host cell DNA. Then the retrovirus starts replication inside a body. Okay, so what is that? Means when this term reverse transcription, this was proposed by Temin and Baltimore in 1970. Okay, in 1970, Temin and Baltimore, they described that reverse transcription occurs in case of retroviral replication inside the eukaryotic cell or inside of any cell. So what happens in this reverse transcription? As here I have told you that this is the single method of genetic information flow. Okay, and here what happens? See, DNA, it replicates, it makes its exact copy. DNA synthesizes mRNA and mRNA synthesizes protein. This is the central dogma. But in case of retroviral infection, the mRNA, that is the RNA of retrovirus, it synthesizes the cDNA, that is complementary DNA, which makes a double stranded DNA. This DNA is the dsDNA or double stranded DNA. Okay, so after discovery of this reverse transcription, many of the causes of cancer, they were clear, and these two scientists, Temin and Baltimore, they also have got the Nobel Prize because of this discovery. Okay, so what happens? Central dogma means it is all about the gene expression inside a cell. How the gene it expresses? Actually, normally gene expression occurs in one-way method. That is, gene synthesizes the RNA, RNA synthesizes the protein. That is transcription, then translation. So this is the one-way flow of genetic information. Okay, so this is central dogma of molecular biology, and if this central dogma of molecular biology is clear to you, then we'll discuss in the next classes about the transcription and translation processes, because already I have taught you the replication. Okay, so if you have any doubt, then comment me, ask me, and like and share my channel for the upcoming videos. Thank you.